In this session, we are going to discuss an important quality topic called validation and verification of laboratory procedures. In laboratories, generating a result is not enough. We must be confident that the result is accurate, reliable, reproducible, and suitable for its intended purpose. Validation and verification are quality activities that help build this confidence. Whether we are performing analytical testing, clinical diagnostics, microbiological analysis, or pharmaceutical quality control, validation and verification ensures that our methods and systems consistently perform as expected. In this session, we will understand the concepts of validation and verification, their differences, performance studies, regulatory expectations, and their importance in maintaining laboratory credibility. Validation provides a high degree of assurance that a process method, instrument, or system consistently produces results that meet predefined acceptance criteria. In simple words, validation answers the question: can this method consistently do what it is supposed to do? Validation is not based on assumptions. It requires documented evidence generated through planned studies and scientific evaluation. Regulatory bodies such as USP, FDA, WHO, and ISO standards expect laboratories to validate critical methods before routine use. Validation, therefore, becomes the scientific proof that a method is reliable and suitable for its intended purpose. This confidence ultimately supports product quality and patient safety. A common misconception in laboratories is that if a method works successfully once, it is automatically acceptable. However, one successful experiment does not prove consistent performance. A single successful run is only a proof of concept. Validation requires repeated testing under different conditions to demonstrate reliability and consistency. According to ICH-Q2 guidances, laboratories must prove that a method is suitable for its intended purposes. The focus is not only on feasibility, but also on repeatability, reproducibility. A method must perform consistently over time with different operators, instruments, and sample conditions. Therefore, one-time success is not validation. Validation requires scientific evidence collected through systematic studies. Many people confuse validation and verification, but they are different activities. Validation is a comprehensive process performed when a new method is introduced or when significant changes are made. It is established performance specifications and demonstrate that the method is fit for purpose. Verification is on the other hand is more focused. It confirms that an already validated method performs properly in a specific laboratory environment. For example, a method validated by a manufacturer may still require verification before implementation in your laboratory. Validation answers: does the method work? Verification answers: does the method work properly in our laboratory? Both activities are essential for ensuring confidence in laboratory results. Validation or verification should be completed before any method, test, or instrument system is used for patient testing or routine laboratory work. This requirement applies to new analytical methods, new instruments, modified procedures, laboratory developed tests, transferred methods. There are no expectations. Even if a method was previously validated elsewhere, verification must be performed before implementation in a new laboratory setting. Another important requirement is record retention. Validation and verification records must be maintained throughout the life of the method and retained for a specified period after discontinuation. Proper documentation ensures inspection readiness and regulatory compliance. Validation involves evaluating several performance characteristics. One important characteristic is bias. Bias measures systematic errors by comparing results against a reference method or accepted standards. Another characteristic is the analyte concentration range. Performance must be demonstrated across the entire concentration range expected during routine testing. This ensures the method works equally well at low, medium, and high concentrations. Validation studies also require an adequate number of specimens. Using more than 40 specimens for comparison studies improves statistical reliability and strengthens scientific conclusions. Other commonly evaluated characteristics may include accuracy, precision, specificity, linearity, and detection limits. Together, these studies establish confidence in method performance. Validation is not limited to analytical methods alone. It applies across many areas of laboratory operations. First, non-standard and laboratory developed methods requires full validation before implementation. Second, modified validated methods must be reassessed whenever significant changes occur. For example, changing reagents, instruments, software, or sample preparation procedures may require revalidation. Third, validation extends to equipment qualification. This includes IQ, installation qualification, confirming equipment is installed correctly; OQ, operational qualification, confirming equipment operates according to specifications; PQ, performance qualification, confirming equipment performs consistently during routine use. Together, these qualification activities support the overall validation program. Successful validation follows a structured approach consisting of three main steps. The first step is plan. Laboratories must determine which performance characteristics need to be evaluated and establish acceptance criteria before starting the study. The second step is execute. Validation studies are performed, data is collected, and results are analyzed scientifically. The third step is document. All procedures, observations, calculations, derivations, and conclusions must be recorded properly. Documentation is extremely important because regulators evaluate not only the study results but also the evidence supporting those results. A well documented validation study demonstrates scientific rigor and regulatory compliance. Large laboratories and pharmaceutical organizations often use a validation master plan, commonly known as a VMP. A VMP serves as the overall strategy document for all validation activities within the organization. It integrates different validation areas such as process validation, cleaning validation, equipment qualification, analytical method validation. The VMP clearly defines responsibilities, schedules, documentation requirements, and compliance expectations. It helps ensure consistency across departments and prevents validation gaps. A well-designed validation master plan improves traceability, audit readiness, and overall quality management. To conclude this session, validation and verification are essential activities for ensuring confidence in laboratory results. Validation establishes that a method is suitable for its intended purpose. Verification confirms that the method performs accordingly within a specific laboratory environment. Both activities must be completed before routine use and supported by proper documentation. Remember three key messages. Validate first. Establish suitable before implementation. Verify before use. Confirm performance in your laboratory. Document continuously. Maintain complete records throughout the method lifecycle. Consistent and documented validation is not simply a regulatory requirement. It is the foundation of laboratory credibility, scientific reliability, and patient safety. Thank you, everyone. If you have any questions, kindly post them in the comment section.